Hello, my name is Nhlamulo, a student at the University of the Witwatersrand. I'll be presenting some of the results from my master's research on the combined effects of cytochrome P415 missense variation. CYPs are pharmacoproteins responsible for processing 75 to 90% of drug molecules, meaning variation in these proteins may be linked to altered drug response. This variation may be described in allele haplotype form, and guidelines have been implemented by groups such as CPIC to better inform prescriptions of certain drugs based on an individual's genetic makeup. However, many of these guidelines may not represent African populations. Therefore, conducting such a study in African populations is crucial in elucidating the precision medicine paradigm in Africa. This leads us to the aim of our study. We want to assess the potential impact of a combination of missense variants within CYP star allele definitions on protein stability and conformational dynamics, two structural bioinformatics approaches for studying differences between mutant and wild-type proteins. Moving on to the methods, the variant data was mined from the pharmacogenomics database PharmVar, and the protein structures were acquired from the Protein Data Bank or PDB. Thereafter, we conducted normal mode analysis combining NCOM and FoldX, comparing the mutant to the wild-type proteins. We then selected a few star alleles enriched in African populations and conducted molecular dynamic simulation using EMBER-20. The system was prepared using TLIP, after which the rest of the process is conducted on the CHPC platform. A two-step minimization was conducted, followed by heating, then simulation at 300 kelvins for 220 nanoseconds. The CPIC guidelines characterize CYP2D6*2 as benign and CYP2D6*17 as having decreased functional effects on the protein. The only difference between the two star alleles is the T107 variant found in *17. It is therefore expected that T107 is the variant responsible for the decreased effect, but our normal mode analysis results suggest that T107 is within a range that is unlikely to have significant effects on protein function. Unexpectedly, STAR2 shows greater variation in folding energy than the decreased function *17, appearing to be driven by R296C. This is figure A. Now moving on to figure B, the RMSIP analysis suggests that STAR2 is more aberrant than *17. This led us to assume that there is an underlying interaction between R296C and T107 that modulates the effects of *17. STAR2 shows the greatest deviation by RMSD from the wild type, followed by R296C. T107 takes longer to stabilize, eventually stabilizing at approximately 16 nanoseconds. PCA of protein trajectories, on the other hand, suggests that T107I dynamics overlap less with that of the wild type compared to R296C. T107 is located within a functionally crucial region of the protein, the BC loop, and it reduces the flexibility of the protein. It, however, has little effect on protein function, meaning it does not act alone in *17. R296C is also located in a functionally crucial region, the I helix. Its effects on protein flexibility have been described to be substrate dependent, which is expected due to the removal of a hydrogen bond accompanied by the mutation. The CPIC star allele functional classification encompasses many other parameters that are not explored in this presentation. It can, however, be said that the observed effect of *17 are partly described by changes in protein conformation and flexibility driven by T107 with apparent modulations by R296C. I would like to thank my funding bodies, my supervisors, as well as the CHPC.